All right, let's dive straight into this explainer. Today, we're talking about the absolute unsung heroes of our biology, ion channels. We're going to explore exactly how these microscopic proteins act as the fundamental link between the outside world and our internal cellular responses. Seriously, by the time we wrap up today, you're going to see how these tiny structures literally dictate everything you do. I mean, think about your grand macro-level human experiences for a second. How exactly does a thought travel through your brain? Or how does a heartbeat maintain its perfect rhythm? What about feeling the sensation of a gentle breeze? We take this stuff for granted, right? But every single one of those experiences fundamentally comes down to the micro-level reality of charged particles moving across your cells. And here's something wild to put it into perspective. Those microscopic electrical signals can travel through your nerves at speeds over 250 miles per hour. No way, right? But it's true. And it all starts with a biological gate. So let's nail down exactly what these gatekeepers are. Basically, ion channels are transmembrane proteins. That just means they span across the cell's membrane to form selective pores. And through these tiny pores, very specific ions, we're talking sodium, potassium, calcium, chloride, and protons, can pass. Now, what makes them so incredibly efficient is that they let ions move down their electrochemical gradient. So they don't directly burn cellular energy or ATP to do this job. They just open the door, and the natural pressure of the gradients pushes the ions through instantly. It's kind of like a molecular water slide.
Section 1, Anatomy of a Gate.
If we zoom way in, every ion channel basically has three core structural components. First up, the membrane-spanning regions. These physically bridge the barrier of the cell's lipid bilayer to create a pathway. Next, we have the selectivity filter. You can think of this like a molecular bouncer. Its entire job is to strictly determine exactly which ions get a VIP pass to go through and which are turned away. And finally, you have the gate itself, which controls whether the channel is actively open, tightly closed, or just temporarily inactivated.
Now, there are 4 major types of these gates, and each one is triggered by specific forces. First, voltage-gated channels. These respond to changes in the membrane's electrical potential, which is absolutely crucial for your neurons and heart. Second, we have ligand-gated channels. These open up when a chemical messenger, like a neurotransmitter, binds to them, driving rapid synaptic communication. Third are mechanically gated channels. These physically respond to mechanical forces, and that's exactly how you experience touch, balance, and hearing. And lastly, leak channels. These are constitutively active, just letting ions move freely back and forth to help maintain the cell's resting state.
Which brings us to Section 2, the electrical spark of life. Well, before a spark can actually fly, a cell needs a baseline, right? A resting membrane potential. In many neurons, this resting potential sits right around -70 millivolts. And this resting state is definitely not an accident. It's carefully maintained by an unequal concentration of ions inside and outside the cell, the constant work of pumps, and, crucially, those potassium leak channels we just mentioned. They allow a steady trickle of positive charge to leave the cell, keeping the inside relatively negative. But when it's time to send a signal, boom, we get an action potential. It's basically this highly coordinated energetic relay race. From the resting state, voltage-gated sodium channels suddenly fly open, and sodium rushes into the cell, depolarizing it. Almost immediately, those sodium gates inactivate, and potassium channels open up. Potassium then rushes out, repolarizing the membrane, until everything eventually settles back down to that resting potential. This super-fast sequential opening and closing of gates is exactly how an electrical signal travels down a nerve fiber. So what happens when that electrical signal actually reaches the end of the nerve? Well, this is where synaptic signaling takes over, and it's a beautifully elegant micro-narrative of biology. The electrical action potential arrives, which triggers voltage-gated calcium channels to open. Calcium rushes in, and that influx forces tiny vesicles to fuse with the membrane and dump chemical neurotransmitters right into the gap between cells. Those chemicals float across, bind to ligand-gated channels on the next cell, and prop them open. Just like that, an electrical signal becomes a chemical one and then immediately becomes electrical again. Let's dive into Section 3, calcium, the master messenger. You know, calcium really is the ultimate intracellular second messenger. Changes in cytoplasmic calcium regulate an astonishing list of functions. We're talking muscle contraction, the release of neurotransmitters and hormones, enzyme activity, gene expression, fertilization, and even apoptosis, which is programmed cell death. But because it has such immense power, cells have to tightly regulate it. Think of it like that strict VIP room again,
A controlled amount is completely necessary, but too much calcium flooding in all at once can be highly toxic to the cell.
To really appreciate this, let's trace the exact downstream signaling pathway. An extracellular signal causes an ion channel to activate. That immediate change in ion concentration, like a sudden spike in calcium, activates second messenger proteins. For example, calcium might bind to a protein called calmodulin. This then activates downstream kinases, which go on to trigger the final cellular response, like a shift in gene expression. It's basically a direct domino effect from the outside environment straight down to the cell's DNA. All right, moving on to section 4, when gates break down. We've been looking at how things work perfectly, but when ion channels are disrupted by genetic mutations, they cause a whole category of severe human diseases known as channelopathies. If a mutation alters how a channel opens, its ion selectivity, or where it's expressed, the results can be absolutely devastating. These include epilepsy from hyperactive neural channels, cardiac arrhythmias from faulty heart channels, certain forms of migraines, cystic fibrosis, and various muscle disorders. Literally, a single broken molecular gate can throw the entire body out of rhythm. And because channel dysfunction is so broadly impactful to human health, ion channels are now major targets for pharmaceutical intervention. They represent the absolute frontier of modern biotechnology and drug discovery. Fixing these microscopic gates is a massive priority for modern medicine today. So, pharmacology uses highly specific drugs to manipulate these broken gates and try to restore normal physiologic function. Depending on the disease, therapeutic drugs are designed to block an overactive channel, activate a sluggish one, alter the timing of its opening, or change how quickly it inactivates. By precisely targeting sodium, calcium, potassium, or chloride channels, scientists can effectively tune the electrical and chemical signaling of our bodies back to normal. Now, you might be wondering, how do scientists even study something so incredibly small and fast? Well, they rely on some pretty advanced research techniques. A vital one to know about is patch clamp electrophysiology. Actually, this technique was so revolutionary, it won the Nobel Prize. It literally allows researchers to isolate a tiny patch of cell membrane and measure the electrical currents flowing through individual, single ion channels in real time. Paired with things like fluorescence imaging, calcium imaging, molecular docking, and cryo-electron microscopy, researchers can literally visualize these invisible gates opening and closing. To fully grasp cellular biology, we really need to understand the fundamental difference between channels and pumps. Channels act as pores, letting ions rush rapidly down their gradient without directly using ATP. They are built for extreme speed and rapid signaling. Pumps, on the other hand, undergo a much slower conformational cycle to physically push ions against their gradient, which absolutely requires direct ATP energy. Simply put, pumps maintain the baseline while channels trigger the action. If you take away just one thing from our explainer today, let it be this. Ion channels act as molecular gates that convert electrical, chemical, and mechanical signals into rapid cellular responses. They are quite literally the translators between the physical universe outside and the biological universe inside of us. Which leaves us with this final thought. If microscopic gates govern every single thought you have, every sensation you feel, and every single beat of your heart, what other secrets of human health are waiting to be unlocked right now at the cellular level? It truly makes you wonder just how much more there is to discover. Thanks for joining me on this explainer, and keep exploring.